Membranes 1. Control molecular traffic across the boundary, includes selective permeability to polar solutes. Organize complex reaction sequences and cell-to-cell communication. Membranes 2. Flexible, self-sealing. Two membranes can fuse. A single membrane can undergo fission. Phosphatidylcholine. A major component of the lipid bilayer of human cells, along with phosphatidylethanolamine. Membrane bilayer. A cell membrane under high magnification. The relative proportions of protein and lipid vary according to the type of membrane. This reflects the diversity of biological function for different cells. Mitochondria. Double lipid bilayer, a lot of undulations, inner, loaded W, a lot of protein where oxidative phosphorylation takes place, outer, makes proteins, more phosphatidylcholine, segregation of molecules based on cell function and organelle function in a cell. Micelle. Individual units are wedge-shaped. Bilayer. Individual units are cylindrical. Vesicle. Has aqueous cavity. Fluid Mosaic Model. Of the structure of biological membranes, outside, glycolipid, oligosaccharide chains of glycoprotein, inside, peripheral protein covalently linked to lipid, integral protein, single transmembrane helix, sterile, peripheral protein, phospholipid polar heads. Biological consequences. Changes in the asymmetric distribution of phospholipids in the bilayer can have biological consequences. Phosphatidylserine is typically in the inner monolayer. If it shifts to the outer layer of an erythrocyte, that cell can play its role in clot formation. In other cells, that shift might signal apoptosis. Fluid Mosaic Model of the structure of biological membranes. Outside, glycolipid, oligosaccharide chains of glycoprotein, inside, phospholipid polar heads. Peripheral proteins, integral protein, single transmembrane helix, peripheral protein covalently linked to lipid, integral protein, multiple transmembrane helices, integral proteins. Membrane-bound proteins that have stretches of amino acids that traverse the lipid bilayer. Peripheral proteins. Membrane-associated proteins that associate with the membrane or with membrane-bound proteins through charge-charge, hydrogen bonding or van der Waals interactions, or those bound to the bilayer with a prosthetic group. Biological regulation. Amphitrophic protein. Peripheral protein. change in pH, chelating agent, urea, carbonate, detergent, integral protein, GPI-linked protein, protein glycan, phospholipase C, integral plasma membrane proteins, classified based on their spatial relationship with the plasma membrane. Bacteria rhodopsin. The most studied transmembrane protein, it is a light-driven proton pump from Halobacterium salinarum. Bacteria rhodopsin characteristics. Membrane-spanning protein, 7 membrane-spanning alpha helices, each helix contains hydrophobic amino acids. The only hydrophobic residues are in the non-transmembrane segments of the protein. Hydropathy plots showing the hydrophobic nature of the amino acid side chains for 2 integral membrane proteins. Top of hydropathy plots. Hydrophobic. Bottom of hydropathy plots. Hydrophilic. TYR and TRP residues of membrane proteins tend to cluster at the water-lipid interface. Porins. Made up of beta strands which traverse the bilayer typically in antiparallel orientation, form channels through which specific molecules or ions can pass. Residues on the outside surface of the barrel are hydrophobic, the channels are under strict control. Membrane proteins with B-barrel structure. FEPA, ample, maltoperin. FEPA. Involved in iron uptake. Ample. A phospholipase, a 12-stranded B-barrel that exists as a dimer. Maltoperin. A maltose transporter, exists as a trimer. Lipid-linked membrane proteins. Covalently attached lipids anchor membrane proteins to the lipid bilayer. Membrane dynamics. Pliable, able to change shape while maintaining its integrity. Individual lipid molecules, membrane bound proteins, etc. also move relative to each other. Varying degrees of disorder. Gel phase or paracrystalline state, liquid ordered state, liquid disordered state. Gel phase or paracrystalline state. Observed at cold temperatures, almost no movement of the lipid molecules. Liquid odorous state. At warm temperatures, molecular motion is evident as well as lateral movement. Liquid disordered state. High enough temperatures where there is near total disorder within the acyl region of the bilayer. Gel to fluid transition. Heat produces thermal motion of side chains. Motion of single phospholipids in a bilayer. uncatalyzed transbilayer, flip-flop, diffusion, uncatalyzed lateral diffusion, catalyzed transbilayer translocation, uncatalyzed transbilayer diffusion, very slow, uncatalyzed lateral diffusion, very fast, catalyzed transbilayer translocation, flipase, flopase, scramblase, flipase, moves P and PS from outer to cytosolic leaflet, flopase, moves phospholipids from cytosolic to outer leaflet, scramblase, moves lipids to either direction, toward equilibrium. 56 milliseconds. Track of a single fluorescent labeled lipid molecule on a cell surface followed for restricted motion of some membrane components. Chloride bicarbonate exchange proteins, glycophorin, anchor and spectrin, path of single lipid molecule, junctional complex actin. Lipids within a monolayer segregate themselves. Glycosphingolipids and cholesterol aggregate away from glycerophospholipids due to more optimized interactions. Sphingolipids typically have long-chain satidecyl groups which pack better with cholesterol. Glycerophospholipids typically have one unsatidecyl group and shorter satidecyl groups than sphingolipids. Raft. Enriched in sphingolipids, cholesterol. Membrane microdomains. Visualized by atomic force microscopy. Membrane fusion. Two membranes must recognize each other, their surfaces must become closely opposed, their bilayers must become locally disrupted, resulting in hemifusion, the fusion of the outer leaflet of each membrane. Their bilayers must fuse to form a continuous bilayer. If part of a mediated or regulated endocytosis, the fusion must be triggered at the right time, usually mediated by fusion proteins. Kinds of membrane fusion events. Budding of vesicles from Golgi complex, exocytosis, endocytosis, fusion of endosome and lysosome. Viral infection, fusion of sperm and egg, fusion of small vacuoles plants, separation of two plasma membranes at cell division. Membrane fusion during neurotransmitter release at a synapse. Neurotransmitter-filled vesicle approaches plasma membrane. V-snare and T-snare bind to each other, slipping up from the amino terminal and drawing the two membranes together. Zipping causes curvature and lateral tension on bilayers, favoring hemifusion between outer leaflets. Hemifusion, inner leaflets of both membranes come into contact. Complete fusion creates a fusion pore. Pore widens. Vesicle contents are released outside cell. Viral cell fusion during infection. Molecular trafficking across a membrane. Simple diffusion, facilitated diffusion, primary active transport, secondary active transport, ion channel. Simple diffusion. Nonpolar compounds only, down concentration gradient. Facilitated diffusion. Down electrochemical gradient. Primary active transport. Against electrochemical gradient. Secondary active transport. Against electrochemical gradient, driven by ion moving down its gradient. Ion channel. Down electrochemical gradient may be gated by a ligand or ion. Simple diffusion 2. The movement of solute particles across a permeable divider from a region of high CONC to a region of lower CONC. Facilitated diffusion, or passive transport. Integral membrane proteins which lower the energy barrier for molecular movement across a membrane by providing a path for the molecules, typically called transporters or permeases. Movement is with the electrochemical gradient. 2 classes: carriers, channels. GLUT1, glucose transporter. Believed to have 12 membrane-spanning helices, side-by-side assembly of several of these helices produce a channel lined with hydrophilic residues which can hydrogen bond the glucose molecule as it passes through the membrane, 50,000 times faster than simple diffusion. T1. Glucose binding site exposed on the outer surface of the plasma membrane. T2. With the binding site exposed on the inner surface. Glucose transport. 1. Glucose and blood plasma binds to a stereospecific site on T1. This lowers the activation energy. 4. To a conformational change from glucose out T1 to glucose in T2, affecting the transmembrane passage of the glucose. 3. Glucose is released from T2 into the cytoplasm, and 4. The transporter returns to the T1 conformation, ready to transport another glucose molecule. Cl-HCO3 exchanger is an example of a cotransport system, found in erythrocytes. Primary role is to increase the CO2 carrying capacity of the blood. 3 general classes of transport systems. Uniport, cotransport, symport, antiport. Active transport. Movement of solute molecules against the electrochemical gradient. Energy is required for this transport. Primary active transport. Transport coupled with an exergonic chemical reaction such as ATP hydrolysis, ATP to ADP by energy. Secondary active transport. Transport coupled with an exergonic flow of a different solute with electrochemical gradient. No K-ATPase. An example of a primary active transporter. Primary active transport. The energy released by ATP hydrolysis drives solute movement against an electrochemical gradient. Secondary active transport. A gradient ion of X, often not, has been established by primary active transport movement of X down. Its electrochemical gradient now provides the energy to drive cotransport of a second solute S against its electrochemical gradient. ATP binding cassette transporters. A large family of ATP-dependent transporters involved in transporting many different kinds of compounds out of cells against concentration gradients, included hero drug transporters. Some have low specificity, others have very high transport specificity. ABC transporters. Found extensively in most all plants, animals and simpler organisms, CFTR and MDR1, most antibiotic resistance is a result of ABC transporters, are good targets for drug design. Secondary active transport. E.coli lactose transporter is one example of the major facilitator superfamily of transporters. Aquaporins provide channels for H2O movement across membranes. Humans have 10 different, and Arabidopsis thaliana has 38 different genes, sometimes appear in a cellular membrane in very high numbers, 2x 10^5 per erythrocyte cell. Aquaporins, AQP1, made up of 4 monomers, each forming a channel through which water molecules can move single file. Facilitate water movement at a rate of 10^9s1. Gated ion channels, Orion selective channels, found in all cell types, used to regulate the cytosolic concentrations of specific ions. Gated ion channels differ from ion transporters in 3 ways. Rate of transfer can be several orders of magnitude faster than transporters. Channels are not saturable. Transporters are. Channels are gated, ligand-gated, voltage-gated. K-channels and Na channels are examples. Both are important in neuron signaling. Nicotinic acetylcholine receptor. Ligand-gated ion channel. The receptor protein binds the signaling compound acetylcholine. This binding causes a conformational change in the receptor protein which opens the gate. Rapid movement of ions, Ca, Na, and Rk into the cell follows, which depolarizes the plasma membrane, triggering muscle contraction. Gated ion channels are the targets of many naturally occurring toxins. In many cases, these toxins are very potent. Underscoring the importance of these channels, some man-made nerve agents also target gated ion. Toxins: VX gas, sarin gas,